Hi everyone, I am Sharks4Kids Ambassador Cailla, and I am here with Jim Gelsleichter from the University of North Florida. So tell us a little bit about yourself.
Hi everyone. Well, my name is Jim Gelsleichter. I am an associate professor of biology here at the University of North Florida. I've been at North Florida for the past 10 years. Prior to that, I was at the Mote Marine Laboratory as a staff scientist for a decade, and prior to that, I was at the Virginia Institute of Marine Science where I received my doctorate for the College of William and Mary, also working on sharks. So I've been working on sharks for close to about 30 years at this point.
All right, so first question we have to ask is, what is your favorite shark?
Well, that's a very common question, and I know that a lot of my colleagues do have favorite sharks, so to speak. To be honest, I never really focused on a favorite shark, but I have some favorites within our survey, and so one of my most favorite species to work with are the scalped hammerheads, and one of the projects that we conduct in our research is we're trying to identify areas that are important to young sharks, and one of the sites that we collect sharks from, we catch a very large quantity of small newborn scalped hammerhead sharks, and I just think they're super cute. They've got big goofy heads and little bodies that are undersized for their heads, and kind of swimming around like like that. Like, what am I doing with this big old head? But it's a really important study because we know that scalped hammerhead populations have been declining. They've been reduced over the past few years, so it's important for us to learn more about that species, and that's one of the ways that we do this.
So you spoke about your survey. What research do you do within this survey here at UNF?
So UNF Shark Biology Program, as we call it, focuses on 3 different areas of research. We do population ecology. In other words, we're out there looking at the species that make up the shark population, particularly within our area, when they come in our area, when they leave, and what areas they inhabit, and what things drive that. And so you can kind of consider that as we're trying to see what sharks are important to Northeast Florida and how Northeast Florida is important to sharks. In addition to that, we also conduct a number of studies on the reproduction of sharks, and so we try to figure out, particularly for different species that we don't know this for, when do they become sexually mature? When do they start reproducing? How many babies do they have? How often do they reproduce? Where do they mate? Where do they give birth? And the type of information that we're trying to figure out is basically how fast that population grows. You need to figure out how fast the population grows so that you can manage it and conserve it. Because, of course, the population growth through birth is the only thing that causes that population to build up. And of course, the population is declining because sharks will die from natural causes, but also from fishing. So if we can figure out how fast it grows, we know how much we can take out by fishing, and how much we have to restrict fishing in order to keep the population growing in a positive manner. And then finally, the last area of research that we focus on is on the effects of pollution on sharks. And so we conduct a number of studies that are geared towards identifying, for example, what was the impact of oil spills, such as the Deepwater Horizon oil spill, which happened in the Gulf in 2010. How did that impact sharks? We test to see how things like plastics in the environment would affect sharks, and environmental pollutants like mercury, for example, and try to come up with a better understanding of whether or not pollution can cause shark populations to decline.
In your research thus far, have you ran into any challenges, or what were your hardest challenges getting to where you are now?
We've got a number of challenges, and those challenges don't go away. It gets harder every year. So our basic survey for shark ecology, there's not too many challenges there. We go out every year. We've been doing it for 10 years, and we set long lines to identify the sharks that live in Northeast Florida, and to look at trends, changes in the abundance of those sharks, whether or not they're increasing, whether they're decreasing. And all that information goes to the federal government so that they can better manage shark populations. And so that's pretty straightforward. We go out and we set long lines, and we've been doing that for a long time, and a lot of people have been doing it for a long time, and that's pretty straightforward. In our reproduction studies, historically, reproduction studies have been conducted, unfortunately, by euthanasia, by killing sharks. And so, in order to figure out all of this information, people would, of course, go out and collect sharks and dissect them. And look at changes in their reproductive organs, whether or not, for example, females are pregnant and have babies, and of course count the number of babies, and try to come up with answers for when are they becoming mature and when are they reproducing and all of those things. One of the things that we try to do is to come up with non-lethal ways to get that same information. And that would include looking at the blood of the sharks. We can collect them and draw blood without killing them and then release them. And to look at changes in different hormones within the blood. We know that some of the hormones in the males, for example, let us know when the males are reproducing. We can use certain things in the blood to tell us, for example, when the females are reproducing. But it's not always consistent from species to species. In addition, we can use an ultrasound to tell whether or not females are pregnant. But again, it doesn't work very well at all times of pregnancy. When a shark is very early in the stage of pregnancy, it's pretty hard to see the embryos. It's hard to count them. And so I think the greatest challenge that we have is to come up with ways to answer questions about the animals that are not in the sort of deliberate fashion that dissection will give you. But it's an important goal that we have because, of course, it's a big, beautiful animal in many cases, and the reality is that euthanizing the animals for some of these studies is somewhat not consistent, I guess, with the goal of conservation, but it's still really important. If we're going to be able to manage the species, we have to figure out how fast they reproduce. But in other cases, for example, small-tooth sawfish is an endangered species. Dusky shark is a prohibited species and is very threatened. In some of these populations, of course, the populations have already been depleted and they're declining, and it is not probably a good idea, and in some cases we're not able to actually kill them and dissect them to get that information. So we've got to come up with other ways to do it, and sometimes that's not as easy and straightforward as it seems. Same thing with pollution. We, of course, are trying to test to see whether or not sharks are affected by pollution. But in order to do that, if we want to do it without killing the animal, we've got to come up with things in the blood or things on the surface of the animal, things that will tell us that the animal is affected by pollution, but not require us to do what would traditionally be done, which would be to euthanize the shark and dissect it and take the liver and measure the amount of pollutants in it and stuff. So, and again, we do a lot of that sort of work with humans, of course, and we know how to do some of that using the blood, but sharks are not humans, and sometimes they make different proteins than humans, and sometimes they respond to pollution in a different manner, and so it's just not that straightforward.
So you're looking for basically a non-lethal approach to the science that's kind of already happening or has already started?
Yeah, so there's a number of non-lethal approaches that have been developed to get at these same questions. And for example, again, trying to get at reproduction, you can use the hormones. But there's a lot of sharks out there, and not all of them, for example, reproduce in the same manner. And so when you look at changes in the hormones, for example, in male sharks, changes in testosterone tell us when male sharks are reproductively active. And that has been shown for most of the different species that we've studied, but there's some species that have weird changes in testosterone. And so what that means is that you can't just universally say, I'm going to study reproduction and I'm just going to look at testosterone in males, because you don't know if the species that you're going to study may differ from the others. And so it's not as easy to just extrapolate from one species, use the techniques that work for one species, and just automatically assume that they're going to work for a different species. Yeah.
All right, well, before we wrap this up, I totally forgot what the last question was. I'm so sorry. What was the last question? The coolest thing you've ever seen. Okay.
Oh, the coolest thing ever.
So before we wrap this up, what is the coolest thing you've seen or learned throughout your years and years of experience working with sharks?
I am still fascinated by the diversity in reproduction in the sharks. And that ranges from what we refer to as oviparity or egg laying. And of course, it's something that occurs in some shark species. They lay eggs, and of course, the baby sharks grow up outside of the mom, and they don't get any extra nourishment from mom aside from the yolk, the egg that they got in the beginning. That's also how skates reproduce. But then we can move towards sharks that are what we refer to as aplacental. That means I don't have a placenta. Viviparous, meaning that they're live bearers. So in other words, they keep their babies inside of them, and the embryos develop inside of mom. But again, all they have is a yolk and egg that they were supplied with. They don't get extra nourishment. But then all of a sudden we can move towards animals that are also aplacental viviparous, but they get extra stuff. And so the stingrays are like that. Stingray babies start off with a yolk, they're inside of mom, but then they use up all the yolk, and then mom starts producing milk from the uterus that nourishes the embryo as it develops in the late stages of pregnancy. And then, of course, a lot of people hear about what is probably the coolest is this ability that some of the sharks, like the sand tiger shark, have, where they also are aplacental viviparous, where they start off with the yolk, they use up the yolk, but then the female the mother starts to produce additional eggs, and they feed on the eggs, and then they may even feed on other embryos inside of the uterus, and undergo what we call uterine cannibalism. And then finally, you get to some of the most advanced species which have a placenta, meaning that they're a lot like humans, where there is an actual blood connection between the developing embryo inside of the mom and the mom itself, so that the blood supply is providing nourishment. And so I think it's just amazing, this very broad diversity of reproductive patterns. It makes my life more difficult, like I said, because all of these different types of reproduction mean that the things that I try to use to figure out the reproductive cycle without killing an animal is often not that straightforward. But it never ceases to amaze me that the sharks have a broader range of reproductive biology or reproductive traits than probably just about any other group out there. And in many cases, like I said, having a placenta, that's not that different than ourselves. So definitely cool stuff.
Yeah, sharks are pretty cool. All right, well, thank you so much, Jimmy G, for everything you've told us today, and then thank you also for all the research you conduct to help save the sharks.
Great, thanks. Take care.